Hello everyone, welcome back to our platform AgriAddict. I am Tulasi, your educator for the subject entomology and nematology. As a part of nematology lecture series, today's lecture will be on nematode management. Up to here, you are well aware of the concepts starting from the history of nematodes, its morphology, biology, ecology, and even nature of damage. So I guess you are well aware of these concepts by this time. Now it becomes an important to learn about the nematode management. This becomes the other aspect of nematology, which is very important. And I request you people to be active throughout the entire session. So let us start our today's lecture without delay. And extent of damage, it is directly related to the population of the nematodes. That means if the population is more, then the damage will be more. That is direct loss to the crop in terms of yield will be more if the population of the nematode is more. Then one more thing we have to keep in mind while we going for any kinds of management methods is we have to maintain the nematode population below the economic threshold level. Why? Because there is a shift in the word from control to the management. What is the difference between control and management? Control is nothing but complete eradication. Management is nothing but maintaining a population below economic threshold level. This plays a major important role in management of any kinds of organisms. It may be insects, nematodes, fungi, bacteria. This concept plays an important role instead of control, because 100% control of anything cannot be possible. So we will go for management methods instead of control, instead of the term control. And even we know that the nematode, when it reaches to the threshold level, or which is also called as pathogenic levels, from here the economic damage to the plant starts. So hence we have to apply the management methods before the nematode reaching to this threshold level or pathogenic levels. And the management measures which are to be adopted should be very profitable and also cost effective. This is the simple statement which is very easily understandable. The whatever we are applying the management methods, it should be profitable in nature and also cost effective in nature, in order to prevent the loss which is that is afforded by the farmer. And the different types of methods or management methods in case of nematodes are regulatory or legal methods, cultural methods, physical methods, biological methods, and chemical methods. So let us know in detail about these types of methods in today's lecture. Regulatory or legal methods. I suggest you people to understand the term or understand the meaning of term or any word before going into the concept. For example, I had explained in the class like ecology of nematodes or nature of damage nematodes were endoparasitic nematodes. Endo is nothing but inside. Parasitic is it causes parasitic when by entering inside the host body. Like this, if we go on splitting the terms and understanding the meaning of the word, then it becomes very easy for you people to understand the concept also. Regulatory or legal methods. Here, regulatory in the sense by applying strict regulations, rules and regulations in order to prevent the spread of nematode diseases or pests from one place to another place. Here in this method, legal enforcement of measures can be applied to prevent the spread of diseases or pests from one place to another place. This is nothing but regulatory or legal methods. The principle mainly involved in the applying quarantine is exclusion of nematodes. That means if the nematode is not present in an area, it has to be completely excluded in order to make it entry of such type of nematodes from the infested area. So these rules and regulations will obey such type of principles. And it also avoids the spread of the nematodes by applying certain quarantine principles, and also traditionally employ some of the methods in order to restrict the movement of infected plant materials and also contaminated soil into a state or country. Which means if a new area, it is not infested with any pests and diseases, here we will consider that is if it is not infested with nematodes. In such cases, entry of these organisms, or there will be some rules and regulations for the movement of any commodities, like it may be fruits or vegetables, from this infested area to the other areas which doesn't have infestation. By having certain rules and regulations, we can able to prevent such type of pests and diseases in the other new areas which is not infested earlier. And many countries maintain certain elaborative organizations. Certain organizations are present in several countries in order to intercept the plant shipments which contains the nematodes and other pests. That means if there are several countries which are having certain rules and regulations in order to prevent, or in order to the entry of pests and diseases through the planting material. Diseased and contaminated plant material, if it is present, they should be treated by applying certain methods like vapor heat treatment and steam treatment. Like these conditions are like these methods will be applied to prevent the entry of such type of pests and diseases, and by this they can be avoided. And also if we found in a lot, if we found any one single pest or any single diseased fruits or vegetables, the entire lot will be rejected, as in case of mainly internationally quarantine methods. We will see here external or international quarantine methods in next slides. Quarantine also prevent the movement of infected plant and soil move out of the other nematode free areas. Quarantine, the best example what I can give is during corona times, that is during the year 2020, movement of individual, that is person, from one place to another place is prohibited. Why? Because as it is spreading from one person to another person, in order to prevent the spread of in order to prevent the further spread, the movement is restricted. So in a similar manner, in case of plants also, movement of planting material is prohibited if it is infested in an area, from that area to another area which is not infested. Different countries have their own priorities, and the legislations have been accordingly, they have been described, like external or international quarantine, internal or domestic quarantine. External or international quarantine, mainly it prevents the entry of exotic pests. That means if in India, if it doesn't have any pest, then the entry of certain nematode pests or nematode diseases can be prevented from other foreign countries. Like this, we can prevent the entry of exotic pest. For example, there is an absence of nematode in India, that is Datilancus destructor, which mainly infests the potato. This can be prevented from entering from other foreign countries to the India. And even these international quarantine methods, they will go for zero tolerance level, which means while exporting any kinds of planting materials or food commodities, they entirely go for zero tolerance, which means the entire lot should be completely free from pests and diseases. If they found any single fruits or single vegetables infested with the nematodes or diseases, the entire lot will be rejected. So export of such commodities will need to be carefully done. And next method is the next type is internal or domestic quarantine. Here the domestic quarantine, which is nothing but within the country itself. within the country, restrictions can be done for the movement of planting materials or for the movement of any kind of fruits and vegetables if it is infested with the pests and diseases. For example, in Nilgiris, which is very prone to potato cyst nematode, that is Globodora roastochinensis, from the Nilgiri areas, that is from Tamil Nadu, there is a restriction for the movement of tubers, that is potato tubers, from Nilgiri area to other states of our country. Like this, the prevention can be made within the country. And there are certain acts which comes under the plant quarantine in India, that is the Destructive Insect Pest Act, that is DIPA, which was enacted in the year 1914, and it was passed by the government of India, which mainly as restriction for the entry of or introduction of exotic pests and diseases into our country from the foreign countries. And there are again some of the agricultural pest and diseases acts which are operated within the states itself. That means from movement from state to state, in order to prevent the movement of or spread of pests and diseases within the country from state to state, there are again certain acts which comes under this quarantine, like Madras Agricultural Pest Act, which was enacted in the year 1919. Within the state or interstates also, these types of rules and regulations are there. And these rules also permit the plant protection advisor for the which will be to the government of India, and he can also undertake the inspection and treatments. Here this means under this act there are some rules and regulations, as I said. According to that rule, plant protection advisor to the government of India who will be present, he'll be having right to undertake inspection and treatments at any times and at any cost of time. And even there are in our country there are strict regulations which have been against the Globodora roastochinensis, which is mainly spreading through potato tubers, that is potato cyst nematodes, and Radinapilincus cocophilus, red ring nematode of coconut. Domestic quarantine regulations have also been imposed in order to restrict the movement of potato, as for both for seed and table purposes. As I explained earlier, the movement of potato cyst nematode from the Nilgiris is mainly prohibited to other states of country, because Nilgiri is very more prone to the potato cyst nematode. So in order to prevent the entry of this potato cyst nematode to other states, certain rules and regulations, that is quarantine regulations, are made. This is about the regulatory methods. Next is the cultural methods. Here the cultural method is the most economically feasible methods, and if these methods are applied in very efficient manner, definitely we can achieve nematode management in very efficient manner also. Cultural control mainly are agronomical practices which are employed in order to minimize the nematode problem in the crops. Like certain agronomical practices will be applied in order to control the nematodes problem in the crops. It may be plowing, like starting from the plowing to the sowing, and also after planting also, like crop rotation, trap cropping, use of antagonistic crops. Like these methods will be used in order to control the nematode population. Let us see the different types of cultural methods which can be applied for the nematode control. Selection of healthy seed planting material. This becomes very important when we go for nematode management, because the nematodes, as we know that some of the nematodes can easily spread from one planting material to another planting material, also from one seed to another seed, because these survives in the planting material and easily they can be entered into those planting material. If you sow the infested seedlings or infested seeds, definitely it will enter into the other cropping season. So such type of in order to prevent such type of infestation, we can go for selection of healthy seedling material. In case of golden nematode of potato, burrowing nematode and spiral, that is Helicotylincus, and lesion nematode Pratylincus of banana, these can be eliminated by selecting nematodes free planting material, because these nematodes, as they survive through, as they enter through the planting material, so selection of LD seedling materials thus prevents the entry of such type of nematodes. And even in case of wheat seed gall nematode, here the galls are formed instead of seeds, and these galled seeds, if we go for sowing, definitely the infestation will be there and it may enters into the other cropping season. So by carefully choosing of seeds which are free from nematodes is most important in such cases. And in case of rice white tip nematode also, this need to be carefully done, which means that a selection of seeds which are free from nematodes should be done. And next is adjusting the time of planting. Here, as the nematode life cycle depends on the climatic factors, we can go for early sowing or late sowing. For example, in case of Heterodora aveni, we can go for fortnight early sowing of wheat seeds, because whenever we go for early sowing of wheat seedlings, as the cyst as the cyst contains eggs inside the eggs inside the body, and hatching of those cysts or hatching of the eggs will takes place very late. So by the time when the plant has emerged and it is now able to sufficient tolerate the nematode population, by that and easily it can escape from the nematode damage very easily. And the other plants, like we can make out like sowing of seeds in during cold season, because during this season the most cyst nematodes will be not much active. So by planting of seedlings in these conditions, that is mainly early potatoes and sugar beets in this cold season, will escape from the cyst nematode during the cold season. By adjusting like this time of sowing, we can go for escape of nematodes very easily. And the next method is following. Following is nothing but leaving the field without cultivation of crops for some period of time. That means nematodes, we know that they are obligatory in nature, which means they cannot survive without the crop or without the host and moisture for even for a few weeks or also few weeks. So after plowing, if we leave the field without cultivation, that exposes the nematode to the sunlight, and even the nematodes, as it doesn't constitute of any crops or cultivation is there on the field, nematodes will die due to the starvation as without the host plant. Hence, by using these methods, nematodes can be easily killed. But this method is not economical, because nowadays we are going for intense farming. Why people will go for following methods? So this method is not economical in nature. And next method is deep summer plowing. Here, deep summer plowing in the sense when during the summer season, the infested field which is infested with the nematode has to be plowed by using disc plow. And by plowing, it may expose us some of the resting stage which are present in the soil to the soil high temperature, and hence the nematodes can be killed very easily. And for example, there is again one method in nursery. We can go for rising of the beds of vegetable crops like tomato and brinjal. During the summer, it has to be covered with a polythene sheet, which enhances the soil temperature by 5 to 10 degrees Celsius, which can kill the nematodes in the seed bed. That means in nursery beds, if we go for plastic, covering the soil with plastic polythene, that is we can go the transparent black. Usually they will prefer for black or white, and it should be very transparent in nature, and usually 25 to 30 centimeter. And if we go for plastic mulching, it may increases the soil temperature by 5 to 10 degrees Celsius, and also kills the nematodes in the seed bed whichever present in the soil. How the mechanism actually takes place is, from the sun, longer wavelengths radiation will be emitted. We know that. To the soil, longer wavelength radiations will be fall. And from after falling into the ground or ground surface, the shorter wavelengths radiation will be trapped inside, so that latent heat will be generated in more. Hence, the soil temperature will be increased more times as compared to normal days without the polythene sheet. Like this, if we go for applying of polythene sheets or covering the polythene sheets on the soil surface, it can increase the soil temperature, and thus the nematode management can be effectively followed. And even this can be efficient. This can be followed in very efficient manner by increasing or by giving irrigation. By giving irrigation along with the soil solarization and even the covering of plastic mulches or polythene sheets on the soil surface, very efficient management of nematodes can be possible. Because irrigation, we know that it also increases heat. So by using deep summer plowing and also the concept of soil solarization along with use of polythene sheet will thus reduce the nematode population very effectively. Next is manuring. Manuring is nothing but addition of certain amount of FYM, that is farmyard manures, oil cakes of neem, castor and press mud, and also poultry manure into the soil, or use of certain green manure crops in or growing of such type of crops in the field will enriches the soil, and indirectly it increases indirectly it will control the nematode by encouraging the development of some of the predaceous nematodes like Monanchus species, which in turn controls the nematode population. By addition of such kinds of manures, it will enriches the soil particles or enriches the soil, and also it encourages the growth of certain antagonistic microbes which will aid in the control of nematode management. And next is flooding. Flooding is nothing but submerged conditions which normally followed in dries. You can see in the pictures. This is the submerged condition. What will happen if you go for flooding? If you go for flooding, then nematodes is not able to take off the oxygen from the soil easily. So this leads to a condition called as asphyxiation, lack of oxygen due to anaerobic condition. So this may create a nematode asphyxiation condition, which leads to the death of the nematodes due to lack of oxygen. And even in flooded conditions, there are chemicals which will be released, and these chemicals are very lethal to the nematodes. Such examples are hydrogen sulfide and ammonia, which will be released in the flooded condition, and thereby it kills the nematodes. So through flooding, we can go if we go for flooding, due to lack of oxygen, there is under anaerobic condition, asphyxiation is created, and due to lack of oxygen, nematode is not able to respire very easily, and hence the death of the nematodes can be possible. And next method is trap cropping. Trap cropping, here one will be the main crop, the other will be used as a trap crop. Trap crop will be very susceptible to the nematode. That means very susceptible to the nematode crops will be grown, taken as a trap crops and grown along with the main crop. Here the crop which is most susceptible will be gradually attacked by the nematodes, and thereby it grows very rapidly in those crops. And after certain period of time, we have to remove those plants which is infested with the nematodes, and it is burnt later. Thus, here the main crop is escapes from the nematode damage. Normally, we saw the crop main crop trap crops we can use in nematode management is cowpea, which is highly susceptible to the nematode damage. And once the cowpea is infested with the nematode, it is removed and burnt. So planning of such type of trap crops should be carefully done in case of nematode management. And also this is highly, as I said, that cowpea is highly susceptible mainly for root knot nematode. So this for the management of root knot nematode, we can go for trap cropping by using cowpea. And the next method is use of antagonistic crops. Antagonistic, which means against nematode. Here these crops are used to repel or kill the nematodes, because these types of plants will be in the roots exudates of these plants mainly contain certain allelochemicals which will affect the nematode population or which will kill the nematodes. By growing certain types of crops in the field, or in, like, we can go growing of certain types these types of plants in border, and which may attract the nematode, and also which may repel the nematode and thus kill the nematode by secreting some of the chemicals. So let us see what are the examples. Mustard, marigold and neem, and even onion and garlic. These are some of the antagonistic crops which can be grown for the nematode management. And these chemicals or alkaloids that is mainly present in the root exudates, which will repel or suppress the plant parasitic nematodes. In marigold, that is Tagetes species, mainly constitute of alpha-tertinyl or bithenyl compounds which are present throughout the plant, starting from the root to shoot tips. The entire plant consists of this compound, that is alpha-tertinyl or bithenyl compound, which will mainly kill the nematodes. For most efficient management method is growing of marigold crops in the main field. This proved to be very effective method. And another is mustard. Allyl isothiocyanate will be present in the mustard, which are very toxic to the nematodes and kill the nematodes. And in pangola grass, that is pyrocatechol is present. It is also act as a toxic to the nematode and kills the nematodes. And certain types, that is enemy plants, that is antagonistic crops, can be grown along with the main crops, are included in the crop rotation, and thus efficient management of nematodes can be done. Next is removal and destruction of infected plants. Here, first initially, we know that the nematodes are also present in some of the stubbles. Those nematodes, in order to prevent the entry of those nematodes into the other new cropping season, so destruction of stubbles need to be carried out. And even the early detection of infested plants can be carried out, and removal of such detected infested plants to be carried out in order to reduce the nematode further spread. After harvest, the stubble of the infected plants are to be removed, as I said initially. In tobacco, for example, in tobacco, the root system is entirely left in the field after harvest, because here the economic part of tobacco is leaves. So after harvest of economic part leaves, the root system is left again in the field without any removal or without harvesting it. So it serves as an inoculum for the next season. For the next season crop, and this inoculum may spread from one plant to another plant. And also in case of Ditylenchus angustatus, the nematodes remains in the left out stubbles in the field. That is here the stem and bulb nematode, which will infest rice. It will remain in the stubbles for some period of time in the dormant condition. So while harvesting of rice grains, we need to even destroy the stubbles, as it contains the nematode juveniles. So such stubbles are to be removed carefully and also destroyed, and land needs to be plowed in order to expose the soil, because soil also consists of some of the dormant stage of the nematode like cyst. So meanwhile, timely plowing need to be carried out in order to expose such type of resting stage which are present in the soil. Use of resistant varieties. Breeding of resistant varieties is very most important in order to prevent further spread of the nematodes, because if you go for resistant cultivars, there is no need of any any need of chemical methods, as we will normally insist chemical methods for control of nematodes if the damage is very more. So if we go for resistant cultivars, as these plants are very resistant to the nematodes, then the damage can be reduced or there is no damage in such type of varieties. Here I have given some of the crops along which are resistant to the nematodes and the varieties. For example, PNR7, Isar Lalit, NT3 and NT12, which is very resistant to the root knot nematode. These are the tomato varieties. And in chili, Pusa Jwala, Mohini, these are the resistant cultivars of chili which is resistant to root knot nematode. For exam point of view, you have to remember the resistant varieties which are present in different crops and against which type of nematodes. And in potato cyst nematode, for Kufri Swarna and Kufri Giriraj, it is resistant to potato cyst nematode. Next method is physical methods. Physical methods here, heat or hot water treatment or steam sterilization. These methods will be used in order to control the nematodes. Normally, these physical methods will able to kill the nematodes and can be applied very easily in laboratory, because these conditions cannot be operated very easily in the field conditions. So such type of methods are very active or very effective in laboratory conditions only. And here the heat, irradiation, osmotic pressures can also be used for controlling the nematodes. And these physical treatments may be hazardous, because while working with the physical methods, the man who are working under who are going for physical methods may in turn exposed to certain kinds of radiations which are followed in control of nematodes. And the first method is heat treatment of soil. Here the sterilization of the soil can be made by allowing the steam, which is a practice which is followed in the soil. And normally this is followed in greenhouses, seed beds, and also for small area cultivations. Here the steam is used for sterilization of a soil, and this can be only possible in small areas like greenhouses, seed beds. Here, and what all the organisms can be killed by using steam is insects, weed seeds, nematodes, bacteria, and fungi. In some cases, like if you go very in deep, in such cases, perforated iron pipes can be buried in the soil. From perforated pipes, steam will be released into the lower level of soil. If there are any kind of organisms which are residing in very depth of soil, in such cases these can be killed out. The soil surface, while going for steam sterilization, it has to be covered with polythene sheath. Polythene sheath will be covered in order to be in order to make this method very effective. And in the laboratory methods or for pod culture experiments, autoclaves will be used in order to sterilize the soil. And next is hot water treatment of planting material. Planting material that like cobs, tubers, or it may be onion bulbs. In such cases, these planting material will be dipped in the hot water, which will be at the temperature of 50 to 55 degrees Celsius. Time will be around 10 minutes. And before planting, this has to be followed, and later planting has to be carried out. By inserting or by dipping into hot water, if the nematodes are present inside the planting material, will be killed out, because nematodes cannot survive at 50 or 55 degrees Celsius, which will be very high. So these kind of methods can be possible in case of nematodes which are infested through planting materials. Next method is biological methods. Here the biological methods in the sense biocontrol agents will be used. That is living organisms. Normally in a rhizosphere, it consists of parasites, predators and pathogens. It may be parasites, predators and pathogens is to be manipulated. These organisms to be manipulated for the control of plant parasitic nematodes. And even this method can be made very effective by the addition of organic amendments such as farmyard manure, oil cakes, green manure crops, and also press mud. As initially I had explained this concept because it increases the multiplication of the nematodes antagonistic microbes. That is, the organisms which are against the nematode or which controls the nematodes, these types of organisms will be increased due to the addition of such type of amendments like farmyard manure or any cakes like castor cake or groundnut cake. Such type of because such type of increase will be there because of great multiplication which has been happened due to in antagonistic microbes. And in other cases also it may help, like due to organisms which are present in the soil. These organisms are involved in the decomposition of organic matter, or so whatever we are applying, no, like farmyard manures or oil cakes. This decomposition will be taken place by the organisms which are present in the soil. By through decomposition, it emits some of the acids like formic acid, acetic, propionic and butyric acid, which will be released in the soil when we during the microbial decomposition. And also some of the gases like ammonia and hydrogen sulfide gases will also be released in the soil. And thus these gases and acids which are released during the decomposition are very toxic to the nematodes, and thus it will kills the nematode. This is the other indirect form of killing the nematode or managing the nematode through addition of organic amendments. This is the same thing what they have explained. Nematode antagonistic microbes will multiply rapidly due to the addition of organic matter, and because these also and in the other case it also improves the soil conditions and helps the plant to grow well. And these material or organic matter will also provide the nutrition for the crop plants. And coming to the organisms which are involved in biological methods are mainly fungi, bacteria, and some of the predaceous nematodes. Here the fungi is also called as a predaceous fungi, as it act as a predator of nematode. Mainly comes under the order monilials and phycomycetes. There are mainly two types of predaceous activities which is shown by these types of fungi: trapping fungi and endozoic fungi. Trapping fungi mainly traps the nematode. Through formation of some of the constricting rings. Here you can see the trap is formed similar to the constricting ring, and it is a non-adhesive trap. The ring which is formed by the fungi, it surrounds the nematode and it restricts the growth of the nematode and thus kills the nematode, and it acts as a infection to the nematode. Thus, by trapping the nematode, the movement of the nematode, the growth and development of the nematode is inhibited, and also the hyphae which are present in the fungi will may easily trap the nematode. And these also secrete some of the adhesive secretions like some of the adhesive secretions which are very toxic to the nematodes and kills the nematodes. Like this mechanism will happen in trapping fungi. And next is endozoic fungi. Endozoic means inside. It will enter inside the nematode. Endozoic fungi usually enters the nematode mainly by a germ tube. which will is able to penetrate the cuticle of the nematode, mainly from sticky spore which is produced by the fungi. By entering if it is a nematode, the fungi will enter inside the nematode and it starts multiplying inside the nematode and absorbs the contents which are present in the nematode, and also it will make the nematode divide of cellular contents or nutrient contents, and thus the nematode will be died. So after multiplication and after some period of time, these hyphae will emerge from the dead cadaver of the nematode. Example in case of this is Catenaria vermicola, which will mainly attack the sugarcane nematodes. And I forgot to tell the example here, Diclaria candida. This is the example for trapping fungi. And apart from these two types of fungi, there is one more fungi that is Purpureacelium lilacinum. Pacillomyces lilacinum. It acts as a parasitic fungi of nematode, mainly root knot nematodes. It is the most efficient or effective control of the root knot nematode, mainly the exchange of nematodes can be controlled by using this kind of fungi. And the next organism is Pasteuria penitris, which is a bacteria. This is a obligate type of bacteria. Mainly it kills the juvenile stages of root knot nematode and very effective against the root knot, mainly through root knot nematodes in many crops. Pasteuria penetrans, you have to remember this is the obligatory in nature, obligatory pathogen. And one more is Poconia chlamydospora. This is a fungi which is also controls the nematode population. There are mainly three species or three types of fungi or bacteria, which mainly three types of biocontrol agents which mainly controls the nematode. That is Pacillomyces lilacinum, Poconia chlamydospora, and also Pasteuria penetrans, which mainly controls the nematode population and shown very effective in nature. Next, coming to the last method, that is chemical method. It is the most efficient method if you want to go for immediate control. But why? Because in biological cultural methods, it takes very time. That is time lasting process. Those methods are. In case of chemical methods, when we go for applying the nematicides, definitely there will be immediate control. This method is lastly preferred when there is need of immediate control, and also when there is most devastating damage from the nematodes or the nematode population is very high. In such cases, chemical methods should be followed. Because initially we shouldn't go for chemical methods is it may leave some of the residues which will be very hazardous to the human and also to the non-target organisms like predators, parasitites, and also even for plant also some of the chemicals may leave the residues and it may affect the plant growth also. So this method should be followed whenever the nematode population is very high. And in the year Kuhn 1881, coming to the history of chemical method, in the year 1881, Kuhn was the first to use the carbon disulfide chemical for the control of sugar beet nematode, which is mainly infesting the sugar beet in field of Germany. And but this doesn't show any encouraging results for him. Later in the year 1911, Besse used the same chemical carbon disulfide for the control of root knot nematodes. Later, the chemicals like formaldehyde, cyanide, these chemicals were observed to have some of the nematicidal properties, and but these chemicals are having very cost effective, that is cost very costly in nature as they are very highly expensive in nature. And in the year 1919, Matthews used the chemical that is chloropicrin, which is also called as tear gas, against the plant parasitic nematode in England. And in the year 1943, the establishment or best efficacy of 1,3-dichloropropene and 1,2-dichloropropane, that is DD mixture, was reported by Carter. This was one of the effective chemical which can be used for the control of nematodes. And in the year 1945, the scientists from California and Florida of USA has reported the efficacy of ethylene dibromide. Later, after discovery of DD mixture in the year 1944, some of the scientists from the California and Florida has reported the efficacy of the ethylene dibromide. In the same year, this was released as a soil fumigant for the management of nematodes by Dow's Chemical Company of USA. And these two chemicals, that is DD mixture and also ethylene dibromide, has paved way for the chemical control of the nematodes. From the discovery of DD mixture and ethylene dibromide, successful chemical control methods have been started. Let us know some of the properties of the different chemicals which are used for the management of nematodes. First is ethylene dibromide. Its the IUPAC name is 1,2-dibromomethane, and it is a colorless gas, and the gas is non-inflammable. That means it can catch fire or it is flammable. It is available as 83% liquid formulation and also contains 1.2 kg of active ingredient per liter, and also it can also be available as granules, that is 35% granules. So it can be used as soil fumigant also. And it is not, one important thing you have to keep in mind is it is not effective against the cyst nematode, that is Heterodora species. So it is not, this chemical is not used for management of cyst nematode, that is Heterodora species, and also for soil fungi. And crops like onion, garlic and other bulbous crops should not be planted after soil treatment with EDB because it is having some of the phytotoxic effect for such type of crop. Hence, after soil treatment with ethylene dibromide, these crops cannot be grown. Or else we can tell that ethylene dibromide should not be used in growing, should not be used on these types of crops like onion, garlic and other bulbous crops. And its trade name or its available form are bromofume or dowfume. This may be asked in exam, like trade name of ethylene dibromide or trade name of any other chemicals. And the next method, the next type is the dibromochloroPropane, and its chemical name is 1,2-dibromo-3-chloropropane. It is a straw colored liquid and weighs around 1.7 kg. It is also used as a soil treatment before planting. And here the certain crops like tobacco and potato, which are very sensitive to the high bromine content, will be very sensitive to the high bromine content. So these chemicals should not be used in such type of crops. It functions very efficiently when compared to other fumigants like ethylene bromide, even at high temperature and high boiling point. Its trade name is Nemagon and Fumazone. These I had left some of the points in between, which is very which can be easily understandable and self-explanatory. Only the important points I am explaining here. And next is DD mixture, and it is a trade name of the mixture of the compounds which mainly consists of cis and trans isomers of 1,3-dichloropropene and 1,2-dichloropropane in 30.35%, and also other chlorinated compounds will be around 5%. And out of cis and trans isomers, here the trans isomer will be more hazardous. That means it is twice the toxic as cis isomer. And it also exhibits some of the phytotoxic effect. What are the phytotoxic effect? Like if it DD mixture is applied on the plant, it shows some of the chlorosis symptoms like chlorosis, yellowing, browning. Such type of symptoms can be seen on the plant. So hence carefully we have to go for using of these chemicals. And its trade name is Tilon. If it consists of only dichloropropene, and if it is combined with a mixture that is dibromomethane, then its trade name is Dorlane. And next is methyl bromide or bromomethane. This is at ordinary temperature, it will be in gaseous form, and also it can be available in liquid form if it is confined in the containers under pressure. And its insecticidal property was first time given by Goupil in the year 1932. And the next chemical compound is 4-8, which is also a insecticide and nematicide. Insecticide, it is in the form of systemic, that is systemic insecticide cum nematicide, which is available in the form of 10% granule. Normally we will go for 4-8 application in control of nematodes, and its trade name is Thymate. Next is Alicar. Trade name is Temec, and it is also a systemic in nature, and it is available form is 10% granule, which can be go for soil application. And the residues which will remain in the plants is for about 30 to 35 days, which will stay in the lethal form. So using of this chemical is most commonly prevented, or carefully we have to use such type of chemicals, and we can go for concept like waiting periods, because as they remain in the soil for about 30 to 35 days, after some period only we have to go for harvesting of such type of plants or commodities. It also acts as repellent, contact, nematicide, and interferes with the reproduction of the nematodes, as it causes sex reversal mechanism in some type of nematodes. Next is carbofuran, which its trade name is Furadon, and it is also a systemic insecticide cum nematicide, and it is available in the form of 3% granule and also as 40F, and the residual effect lasts for around 30 to 60 days. It is more than the Alicar. And here the systemic chemical has got acropetal action, which means from the site of action, it moves upward through xylem. And it is applied at the rate of 1 to 2 kg active ingredient per hectare. Normally, when we go for nematode management, the most chemicals which we'll be using are 4-8, carbofuran, and even Alicar. Sometimes we will go for Alicar or even phenamiphos. These are the main frequent chemicals which will be applied for the nematode control. So these are the different management methods while going for the nematode control. So for applying or for getting an effective or efficient management control of the nematodes, instead of going for individual methods, we can go for integrated nematode management by combining all these methods like regulatory, physical, chemical, biological, and even cultural. These methods can be applied in compatible manner or in well coordinated manner. If we go for these type of methods, then it is possible to control the nematodes in very efficient manner. So thus integrated nematode management is followed, or it is recommended to follow for the control of nematodes in order to get efficient management. So in JRF point of view or any exams point of view, you have to remember the different concepts like cultural methods, what cultural methods can be applied for nematodes and some of the antagonistic crops and some of the chemicals which will be used as a nematicide for the control of nematodes, and even for physical controls, what kind of methods comes under physical methods. So this is all about the today's lecture. I hope you have understand the management different management methods. So thank you.